Okay, so here we're going to look at variables as part of my series on the scientific method or the nature of science. So when we look at variables, we're going to use the example from my previous video on hypotheses about aphids and rose bushes. So at the end of that video, we learned a tiny bit about how aphids exist, right? So they're little insects that have these openings called spiracles in their body, and the spiracles are where gas exchange occurs. So that is like the hole or the opening where air comes in, like with oxygen, and then carbon dioxide goes out. And then they travel through trachea, which are little tubes, to deliver that gas, the oxygen, to the tissues in the insect. So anyway, you do some research and you find that a common way to get rid of aphids off your plants is to spray them with an oil-based solution. So you wonder, in your cabinet at home that you have you've seen coconut oil that is solid at at colder temperatures. And you wonder if the consistency of the oil matters as far as part of the effectiveness of getting rid of aphids. So you do some research and you find that the melting point of coconut oil is 24 degrees Celsius. And so you design an experiment to test the effectiveness of removing aphids with coconut oil heated or cooled to different temperatures. So when we think about what is an appropriate hypothesis for this experiment, I have three here. If you remember, a hypothesis is an explanation that you can test. So for part A, the aphids will die if I spray them with cold or thick coconut oil. That's actually like a prediction. And then the next one, spraying the aphids with coconut oil that is at the lowest temperature to reach a liquid state will have the greatest impact on removing aphids. Again, that's also a prediction. There's no explanation there. A hypothesis is an explanation that you can test. Both A and B are both predictions. And then C says, spraying the aphids with coconut oil that is at the lowest temperature to reach a liquid state will have the greatest impact on removing aphids because it'll be the thickest coating of oil across the spiracles. Well, now there's an explanation that you can test. Okay, so let's go ahead and see how we can set up an experiment. So once we've developed our hypothesis, how do we test it, right? So we need to set up a controlled experiment. But before setting up an experiment to test your hypothesis, you need to identify different variables. But what is a variable? Well, a variable is anything that can change or be changed in an experiment. It varies, or things that can vary, I guess you can say. So there are three different kinds of variables that are important to understand. We have our independent variable, our dependent variable, and then our controlled variables. So in the next section, I'm going to talk about independent and dependent, and then we'll end with controlled variables. Okay, so let's go back to the roses and the aphids. Let's say you're setting up a controlled experiment to test the effectiveness of different temperatures of coconut oil. So you plant 3 rose bushes, or you buy 3 rose bushes of equal size. You add 10 aphids to each plant, so you're keeping it nice and even and equal. Okay, and then you assign each plant a different temperature of coconut oil. So we have 25 degrees Celsius, 30 degrees Celsius, and then we have 35 degrees Celsius. Now, you spray each plant with 5 sprays of coconut oil. Okay, and then some of the aphids die, and then you take your observations and you record the number of aphids on each plant. So, in this experiment here, let's talk about our first variable called the independent variable. The independent variable is what the researcher or the scientist is changing or what is being tested. So in this experiment here, I want you to think about what are we testing? What are we curious about? What are we in charge of changing? So here we have 3 choices. Which of the following is the independent variable? Is it the number of aphids at the end? Is it the temperature of coconut oil? Or is it the number of sprays? Good. It's the temperature of coconut oil. That is what we are changing as the researcher. That's what we're curious about. So we are setting that independent variable, and then we're going to count after our experiment, after we spray them with coconut oil, we're going to look at, well, how many aphids are left? How did it work, right? So that brings us to our dependent variable. So our dependent variable is the result of our experiment. It's what we are measuring, what we are counting, what we are recording. Like, what are we checking on? So the dependent variable is based on the independent variable. It depends on the independent variable. So in our experiment, what is, that should say dependent variable. I copy and pasted, darn it. So what is the dependent variable? Scratch that. Oh, maybe I can. Oh, I can't. Yeah, shoot, sorry. It is the number of aphids at the end. So the number of aphids depends on our independent variable. Okay, so here, let's go ahead and look at our third variable, the controlled variables. Not to be confused later with control groups. But anyway, so controlled variables are the things that remain the same between all control and experimental groups. So here we have 3 different groups at 3 different temperatures, but we kept, as much as we could, the same, right? So here, examples in this experiment of controlled variables include the number of aphids. They all had 10. We also had the species of plant and insect. We used aphids and we used rose bushes. We have the amount of water. So there, I mentioned the previous slide that they're all in the same like sprinkler system, so they get the same amount of water. They get the same amount of sunlight, the same amount of sprays, etc. And so these are all controlled variables. We are keeping everything the same except the temperature of the oil. That is the only thing we change between our different groups. So therefore, if we see like different results, we know it's due to temperature and not because one plant got more sun or more water or more sprays. We know that the only difference was the different temperatures, and therefore our results are based solely on that because everything else was equal and controlled. So keeping your controlled variables even and the same are super important between your different control groups. So when I look at this experiment here, after conducting this experiment, we see that there are 7, 6, and 5 aphids on the different experimental groups. Now, you might be thinking, oh, cool, my hypothesis was that the cooler temperatures will be more effective because the coconut oil is thicker. And you see here like, oh, shoot, the 25 degrees Celsius, only 5 aphids remaining. That means it had the most to die. It was the most effective. But let's be real. You had 3 setups with only 10 aphids on each plant. Do you think this is really enough information to draw accurate conclusions that you can say with certainty, yes, my experiment worked and I feel confident in my response? Probably not. The next best thing to do would be, you have 2 choices here, to increase the validity of your experiment, to make it a stronger experiment that your conclusions are more sound and reliable. You have options. Well, really should do both, but you should have a large sample size and multiple trials. So here, I can start with my 3 groups, and then I can increase my number of rose bushes that I plant or that I use. And therefore, I'm increasing the amount of aphids. So when I eventually do, when I do spray them each with the different temperatures, I have more data. It's more convincing of an argument. So for example, if I surveyed 5 people and I said, do you prefer M&Ms versus Skittles? Right? And 3 said they prefer M&Ms and 2 said Skittles. And I'm like, oh, M&Ms are the best. That's not very convincing. I only asked 5 people. That's a very small sample size. First, if I survey our entire high school, which is 4,500 students, and I find that 70% prefer M&Ms over the 30% Skittles, that's a bit more reliable. Right? I have a way larger sample size. It's a more convincing data. Same thing you kind of experience when you buy things on Amazon. You look at the number of reviews. And so when you look at the number, like this camera that I just bought, for the price that I paid, it had the best reviews with the most. It wasn't just 4.5 stars, but it had 72 reviews, which was more convincing to me than the other camera of the same price with 4.5 stars that only had 7 reviews. And so the more data you can collect, the better your experiment's going to be. But now, so here I have large a larger sample to see the effectiveness of different sprays. So that's important. But you know what? You might as well just conduct your experiment all over again just to confirm. The more times you can repeat your experiment to confirm your data, the better. I just watched that radioactivity movie on Amazon about Madame Curie, and she repeated her experiment multiple times on her way to discover a new element. And so, yeah, repeatability is also important to confirm that your results weren't just a fluke. Okay, so let's look at a couple other experiments as examples to practice with these words of independent, dependent, and controlled variables. So here, we know that plants need light for photosynthesis. So you have a prediction that if I plant a tree near light, it will grow better. Now, if we turn that into a hypothesis, it may sound something like, due to photosynthesis requiring light, seeds planted with more light will grow taller when compared to plants grown in less light. I have a bit of an explanation there. There's a reason why I'm saying what I'm saying. So here, if I look at some variables, my independent variable, think to yourself, what is the independent variable? What is the researcher in charge of changing here? And then, so that independent variable, what you change, and that's the amount of light. We have one plant that's close to the window and a plant that's further away. Now, what depends on the amount of light? Like, what are we measuring? What are we observing? It's going to be the growth of the plant, the height of each plant. That is the dependent variable. It depends on how close you put it to light, to the independent. And then our controlled variables, there are factors that we keep the same. So the amount of soil is the same, the size of the pot's the same, the amount of water, the fertilizer, the species of the tree is the same. So everything is the same except for my independent variable that I'm changing, the amount of light. So I know that if there's any difference in height, it's due to light alone and not because I use different fertilizers. So controlled variables or constants are very important. Now let's look at one more. So let's say you're curious about an airplane, a paper airplane. Your big question is, how does size of the paper airplane affect the distance it travels? Like, you're curious, is it better to make smaller paper airplanes or larger paper airplanes if I want it to travel the furthest, right? So your hypothesis, the plane with the largest surface area will come into contact with the most air, and therefore it will travel the farthest. Okay, so you throw your paper airplanes, you make them of different sizes. All right. Let's go ahead and just talk about some, it's not the best hypothesis, I'm sorry, but I thought it was a good example of variables. So here, our independent variable, think to yourself, what's the independent variable here? What are we in charge of changing? Good, it's the size of the airplane, the paper airplane. What's the dependent variable? What are we measuring? We're measuring the distance it traveled, because that's going to be dependent on the size of the plane. And then, what are some controlled variables? Now, this one's a little bit harder in paper airplanes because the force with which you throw it, like, you could have this like bias that you want the larger plane to work, so you throw that one a little bit harder compared to the other two. That's not good science. The best way would be to figure out how to have them all have equal force. So maybe it's like a robotic arm that lets them go, or you put a fan that's on the same speed and you put it in front of the fan and then they go, making sure they're at the right angle or something. So you want to make sure that the only difference is the size of the plane. So the other variables you would keep controlled would be the design. So you'd have to fold them in all the exact same style so you know it's not due to the design of the paper airplane. You would also want to make sure you use the same type of paper. Using cardstock or construction paper or newspaper would have different effects because those are of different weights. And so, this was just another example of variables. Oh, and that's the end of my video on variables. Good job.